how would you, I guess, what do we understand today about what constitutes a healthy microbiome?
Yeah, that's the $1 million question. We still don't know what a healthy microbiome is. And yet it's very easy for us to make claims about what's unhealthy, even as scientists, in diseases and things like that. What we have done, I think, to get around that is we use each person as their own control. So where they started from and where they've moved to given a certain intervention or observation over time. It's really hard to do cross-sectional studies across populations and make conclusions about what's healthy because everyone is so different in terms of their microbiome. So we know what pathogens are. We know that there's C. diff, and we know there's certain bacteria that consistently are involved in disease processes. And if you have an overabundance of them, that may not be a good thing. But in some people, it's completely normal. So it's really hard to tell. But there are, generally we know that we have firmicutes and bacteroides, and those are the main populations, and that that will make up, it's almost like a 50-50 mix, let's say, of those two, and then everything else is sort of smaller populations. And you kind of want to see certain consistencies in your microbiome, but that's such a crude description that if, again, you zoom in, you'll see little blips that could cause alarm in one person and be totally normal in another. So I don't have a good answer for you.
Makes it hard to do microbiome testing then.
Yeah.
And give someone some sort of information they can do anything with.
Yeah, I talk a lot on this topic. Yeah, because it's a tough one. There's so many at-home microbiome tests these days. And I don't think we should stop making these tests or innovating on these tests. I think there's a future for them. I just gave a whole lecture on this a couple weeks ago, but and I won't go into much of the details, but there's not very much transparency.
Is that available online or anything?
No, it's not available online. But I gave a talk at an integrative health.
Well, you can give us
the
synopsis.
Yeah, it's mainly that. Okay, if you were to take your stool sample and send it to 5 different companies, you'll get 5 different results. And the reason is because they're all using different methods of analysis, both in terms of how they sequence and how they analyze the data. And you don't really know often how they're doing it. And that can make a big difference in the data interpretation and what you can expect to get back.
Is there a gold standard?
Again, it depends on the question. So if it's a stool sample, a metagenomic sequencing is kind of what we do as a gold standard. There's still some issues with that as well, but metagenomic sequencing will give you a lot more information,
right. So the bacteria that's in the stool is highly correlated with what's in the colon?
With the colon, yeah, I would say yes, but not anywhere else.
Okay.
Yeah.
Where did the kind of concept or idea of diversity come from? So there's research, Rob Knight's work that has looked at, and I know you'd be familiar with this, but just for the listeners to refresh them, it was looking at the sort of diversity of someone's microbiome and the amount of plants in their diet, and it didn't matter. The type of diet that people were eating didn't matter, the label at all, omnivorous, whatever. But what seemed to associate with diversity was plant diversity in the diet. And a lot has, I guess, been made of that. But I guess underpinning that is first the assumption that diversity is really important. So what does that actually mean, diversity? And is it important?
Yeah. Diversity is probably the one metric in the microbiome field that everyone looks at and everyone kind of points to as more diversity being good and less diversity being bad, or loss of diversity being bad, quote unquote. And yeah, a lot of this was pioneered by Rob Knight's group. And the concept is the more different types of bacteria that you have, the more essential functions can be carried out by your gut. And also there's a lot of redundancy in your microbiome to a degree, and there's nuances there we don't need to go into, but there's redundancy. So if you were to take a new drug or change your diet and it caused a dropout of some of your bacteria, the more diversity you have, theoretically, would give you a better chance of still being able to carry out those essential functions, because not just one bug, one function, that would be evolutionarily unfavorable. And so the reason is that you can still, there's balance where if one bug carrying out that same function drops out, there's another bug that can pick up, the slack. That's kind of where the concept of diversity comes from. It's an evolutionary based term that applies in in mammalian scales, but also in bacterial scales as well.
Yeah, I kind of, whenever I think about that concept, I'm not sure if this is a good analogy, but I think of like a functioning city and having lots of people, a diverse population, different genders and people with different cultural backgrounds, and they're all able to contribute to a better functioning society, environment.
Yeah, that's a great analogy, yeah.